My laboratory is part of the new Center for Research, Innovation, Surveillance in COVID-19 and other health emergencies. Our laboratory, our center is based on 13 laboratories, several multi-user facilities, a BSL-3 facility, and a BSL-3 animal facility. My group of research is in immunopharmacology. So as Dr. Ruth introduced, our laboratory is focused on translational and basic research devoted to the interplay between inflammation, metabolism, and the mechanisms of pathogenesis of infectious disease. We work mostly with emergent diseases like and viral diseases like dengue, Zika, chikungunya, and COVID. But we also work with some other infections, including malaria and sepsis. So our team aims at identifying biomarkers and therapeutic targets for those diseases. So our group has three main aspects of the study. The first one is in the immunopharmacology of COVID, influenza, and arbovirus. So we are interested in the immunometabolic responses, in the determinants of severity and long-term consequences of disease. And today I will talk more on this aspect of immunometabolic regulation in infection. We also work on antimicrobial resistance, neuroinflammation, and long-term consequences in sepsis and malaria. And in terms of repositioning of drugs and new development of drugs with antiviral and anti-inflammatory action. So our group has been interested for quite some time on understanding the immunometabolism and their importance for host pathogen interactions. And that because immune response does require major changes in the metabolic process, and that is necessary for the full activation of immune cells to determine their ability to divide, differentiate, and to carry on effector functions. The metabolic status of immune cells, they go into reprogramming during different conditions and are quite remodeling during infection. And it's important to say that the metabolism of the host cell can be a major determinant for pathogen growth or containment. So in terms of virus, viral as an obligatory intracellular pathogen, they hijack several of the metabolic pathways of the host cell for their entry and replication. And that has been well established for different of the viral pathogens. Today, I will focus my talk on the aspect of and the importance of lipid metabolism for pathogen entry and replication. I will focus more in the aspects of replication and in the inflammatory response. And our group's quite much interested on the roles of intracellular organelle called lipid droplets and how lipid droplets can be hijacked by virus to support their replication within the host cell. So lipid droplets are highly conserved organelles during evolution. They can be observed from even in bacteria exhibit lipid droplets. parasites, unicellular parasites like T. cruzi can have lipid droplets. In mammalian cells, the cells that are professional in inhibiting lipid droplets are the adipocytes, as we can see here, that form large unilocular droplets. But they are also highly regulated in immune cells, as we can see here in macrophages that have upon stimulation at high numbers of lipid droplets within the cell. So lipid droplets, they are cytoplasmic lipid domains. Although they do not have a typical bilayer membrane, they do have a quite unique membrane of specific fatty acids, and more importantly, they do contain a highly dynamic proteomic, and that will be important for their functions within the cell. So lipid droplets, depending on the cell and within the stimulatory conditions, can inhibit different proteins that compartmentalize within the lipid droplet, and that confers different functions to this organ. So different groups have established quite a number of proteins, including enzymes involved in energy and lipid traffic and energy homeostasis, as well as in lipolysis. And for our interest, another aspect that's very important in lipid droplets in immune cells is that they can be also very important hubs for inflammatory mediator production. So they do exhibit upon stimulation a number of enzymes involved in lipid mediator synthesis, as well as different enzymes involved in signaling, like PI3 kinase. MAP kinases can compartmentalize within the lipid droplet phospholipase A2, as well as more recently, it has been established quite an important role for lipid droplets as an immune hub that can compartmentalize different proteins involved in interferon synthesis, in antiviral synthesis proteins like piperine, as well as antibacterial proteins that can compartmentalize there, and other cytokines and enzymes important for the immune response. In addition, lipid droplets functions are very important in terms of cellular organelle communication. So their function is also very highly regulated upon organelle interaction. So as we can see here, lipid droplets are in close contact with the cytoskeleton, and this way they can move into the cell. They can establish contacts with mitochondria and regulate metabolic functions within the cell. Also, lipid droplets, they are formed after a very close contact with the ER, and mostly conditions, they keep in touch with the ER, and there are quite a number of exchange of molecules between the ER and the lipid droplets. And also lipid droplets establish contact with phagosomes containing bacteria and other pathogens, and I will talk more about today on viral replication compartments and how lipid droplets can contribute to that as well. All these connections are very highly regulated and depend on the interactions of very specific proteins within the droplet and within the organelle. So although they do not have a typical bilayer membrane, they do establish contact and transfer regulated, molecularly regulated transfer of content between organelles. So lipid droplets has been established as an inflammatory organelle, and work from our group and other groups for quite some time, has established that increased lipid droplets in size and numbers are observed in inflammatory diseases that include atherosclerosis, tuberculosis, sepsis, and many others. They are vitally distributed, inactivated, but not resting inflammatory cells. So in most cases, macrophages at resting do not exhibit lipid droplets, but that can be highly rapidly induced upon different stimuli, here with hot cell gel or here with LPS stimulation. And we can see that increase in different cells of the immune response, including macrophages, neutrophils, endothelial and epithelial cells. And in these circumstances of activation, they are highly involved in the heightened production of inflammatory mediators, contributing to the inflammatory response. So more recently, we also established together with Albert Poe and Robert Pathogen, an important role for lipid droplets in the signaling involved in interferon signaling and antibacterial host defense mechanisms, showing that lipid droplets can interact directly with bacteria and transfer antibacterial proteins, leading to the control of bacterial infection. And also, there are quite some interactions with mitochondria that would lead to an important role in lipid droplets regulating the metabolic response of the inflammatory cells and contributing to this regulation that's quite important for the full activation of macrophages and other cells, for instance. And interestingly, lipid droplets are quite highly regulated with different infections, and that include bacterial, viral, fungal, and parasitical infections. This regulation is highly regulated. It involves different innate immune receptors that through very specific signaling pathways, including transcriptional regulated pathways, that most important PPAR, gamma, and that RBP participates quite importantly in regulating the lipogenesis that leads to lipid droplets formation upon infection, but also it involves a control of the lipolysis of the cell, and as well as the interaction with mitochondria. They all will depend on the balance that will lead to increased lipid droplet accumulation in the cell. So lipid droplets, and our group has been studying the functions of lipid droplets in different infectious disease. And our view of lipid droplets during infections that pathogen-induced regulated biogenesis, and that is highly regulated. And they have, although they have evolutionary conserved functions in host defense, intracellular pathogens, and specifically highly intracellular pathogens that can hijack lipid droplets for their survival and replication. They can act as signaling platforms for inflammatory mediator production, but they also have roles in this amplification of inflammation that can contribute to organ dysfunction and severe disease. So today I will present to you the work on two different areas. The first is on Zika. So some of our recent data on Zika-induced modulation of lipid metabolism in neurocells and their functions of lipid droplets in Zika virus replication and neuropathogenesis infection. And afterwards, I will show you also discuss with you some of the work on the modulation and functions of lipid droplets in SARS-CoV-2 infection. So we've been working with flavivirus for quite some time, and that the interaction with lipid droplets, we start working with dengue virus infection, and that's a lot based on the work of other groups that have established before an important role for lipid droplets in HCV infection. So in dengue studies, we've shown that lipid droplets are increased in the cells from patients with severe dengue infection, and that lipid proteins from dengue virus, the capsid protein of dengue virus can interact with the lipid droplets, and this will be so, and this interaction, if you disrupt the interaction of dengue virus protein with lipid droplets that have major roles on disrupting replication of dengue virus, and you can also do that by inhibiting lipid droplet formation within the cell that would also have major roles in dengue virus replication. So keeping that in mind, we wanted to investigate that during Zika virus infection, we would have similar roles for lipid droplets in the Zika virus replication, especially looking into neurocells. So Zika virus is transmitted by infected mosquito in tropical and subtropical areas. Zika is also transmitted from mother to fetus during pregnancy, and that has been quite important in terms of their pathogenesis. In Brazil, we had an important outbreak of Zika that during pregnancy, infants were born with microcephaly and other congenital malformations, as well as preterm birth and miscarriage. Zika virus is also associated with Guillain-Barré syndrome, neuropathy, and myelitis in adults and children. So not only during development, Zika virus infection would be important in terms of neuro infection. So in some recent studies from different groups has established that lipid droplets can accumulate in the central nervous system, and in that case, it can participate in the pathogenesis of different diseases, and that participates in neurodegeneration. And one aspect of this pathogenic mechanism was by the accumulation of peroxide lipids, and lipid droplets would participate in diseases, neurodegenerative diseases. However, the roles of lipid droplets during infection of the central nervous system have not been established. So we went on to carry out experiments to see if Zika infection of neural cells would have roles in triggering lipid droplet formation, and how would that outcome be for viral replication and also for the cell. So we use different human neurocell model. One is the neural stem cells derived from iPS. And we can see here that Zika-induced infection leads to increase or to a different reprogramming of the lipid metabolism of the cell, inducing activation of the transcriptional factor SRBP, as well as increased expression of PPAR gamma. And we went on to demonstrate that indeed Zika can also regulate the lipolysis within human neural cells, as we can see here by regulation of ATGL and HSL. And as we can see here, lipid droplets both in human neural cell lineage or in neural stem cell can trigger quite an impressive accumulation of lipid droplets that we can see here in red in the infected cells when compared to the control or to the non-infected cells. We went on to see if we inhibit lipid droplet formation, and we use that by inhibiting a critical enzyme that works in the last step of triglyceride synthesis, that's the DGAT1. By using the inhibitor of DGAT1, we can almost completely block the lipid droplet accumulation within the cell, and we can see here quantifying the inhibition of lipid droplets, and we went on to see if that would have impact for viral replication, and that's what we see here in this figure, that by PFU we can see that when we inhibit lipid droplet formation, we see a quite dramatic reduction in viral replication, as well as we can look for double-strand RNA staining as sites of viral replication within the cell. We can also see a dramatic reduction in the viral sites of replication in the cells that were treated with the inhibitor of DGAT1. And we went on to establish on in vivo experimental models. So the animals were infected at P3, and after different times of infection, we looked for viral replication in the brain, the inflammatory response, and also of lipid droplet formation. So first looking at P13, we can see that indeed lipid droplet inhibition by the DGAT1 inhibitor leads to a dramatic reduction of viral loads in the brain of those animals. And as we can appreciate here, indeed, the animals that were treated with the inhibitor of DGAT1 exhibit a higher survival rate when was compared to the animals that were infected but not treated. So we went on to see in tests of to see if lipid droplet inhibition would have roles in preventing long-standing cognitive impairment due to Zika infection. In this case, the behavioral tests were performed at P60. At this time, we do not see any virus in the brain anymore, but we at P60, we don't see virus in the brain anymore, but using different tests for neurocognitive response, we can see quite a high protection on the neurocognitive response in the animals that were treated with the inhibitor of the GAD when compared to non-treated animals. Also, the inflammatory response was quite regulated, and we can see that pro-inflammatory proteins were inhibited in the cells that have the lipid metabolism inhibited. And as we can see here, this is an in vivo microscopy of the brain, and we can see the increased lipid droplets in the brain of animals that were infected with Zika virus, and that was reduced by the treatment of the inhibitor of DGAT, and also we'll be able to make the see. Yeah. So we can see that in Zika virus infection, the increasing in adhesion and rolling of the cells in the in vivo microscopy, and we can see that animals that were treated with the DGAT1 inhibitor, there was an inhibition of the neuroinflammation in the cell. And so we went on to see if this metabolic reprogram were involved in triggering lipid peroxidation in the brain. And the answer is yes. We went to see different lipid peroxidation metabolites, including MTA, hydroprostane, and for hydroxynonenal, and we can see that the treatment decreases the lipid peroxidation that can be a cause of the increased damage to the brain. So, we've been studying mostly in RNA positive virus. However, there are studies now with different viral classes, including DNA virus that also needs lipid droplets for replication, and also influenza virus and other virus. The mechanism for each virus, it varies. So which component of the interaction most times are different for each virus, how they explore this interaction for viral replication. But it's been established an importance of lipid droplets in different viral.
Okay, so maybe it's lipid droplets mainly affect the virus replication factory inside. So maybe some viruses, they are replicated in the nucleus. They are very different mechanisms.
It may occur in different mechanisms. Well, there are some groups that I'm not working with that, but there are also lipid droplets within the nuclei of the cell. So not sure if that would have any role, but depending on the virus, it can work in different aspects, like I showed in the SARS-CoV-2. In great part is for contributing to the viral replication compartment. In dengue virus, the mechanisms are different for enabling RNA replication and also in viral particle formation. So it may occur in different steps in the cell. And that depends molecularly for each viral. The interactions are different. Even the proteins from the viral that interact with the lipid droplets, they may be different for each virus, like dengue and HCV, that although they share lots of common aspects in their replication, the mechanism of interaction and are different from dengue virus for HCV virus.
Thank you.
Yeah, yes.
So, Patricia, if I understood correctly, is this drug that you use to inhibit the enzyme been approved for use in humans? And so the question is, could you use this inhibitor as a therapy for and to show for controlling infection by different sorts of virus?
Yeah, so this drug has been developed for obesity treatment, but there were some clinical studies, but it was stopped during the obesity view because of their collateral effects in mostly diarrhea and other mechanisms of lipid reabsorption in the intestine. Has been some studies with HCV and this drug. The idea here is more on thinking, not necessarily for this drug as a repurposing, but more on thinking that targeting some of the host cell can be a way of inhibiting different virus for a antiviral therapy so that you can get prepared for a drug that can inhibit by targeting the host cell and mechanisms that are very important for the viral replication, since the viral would need to hijack all the metabolic system of the cell for their replication. So that would be a general mechanism to go for a broad antiviral. So the idea, I would say, in the step that we are now, it's more in terms of the concept of using this as a target more than this inhibitor itself. So maybe other inhibitors may be advantageous, but this one has passed through phase 1 and phase 2 trials. So maybe it is a potential target, the drug itself, but I would say thinking as a lead to develop other compounds maybe also